Microscopic test.
Habitat of Streptococcus pyogenes. They are found in throat and skin of humans. They are commensals of upper respiratory tract. Carry rate of Streptococcus pyogenes in the respiratory tract is only few in adults, over 10% in children attending kindergarten. Morphology of Streptococcus pyogenes. Number 1, gram plus V bacteria. Number 2, spherical or oval cocci in chains. Number 3, 5 to 1 in diameter. Number 4, non-motile. Number 5, non-sporing. Streptococcus agalactiae is a part of the human normal flora and colonized area like the gastrointestinal tract and the genitourinary tract of post-adult, but it is also associated with severe invasive infection, mostly in neonates, pregnant women, older adults, and other immunocompromised individuals. It is considered a pantobioint that converts from the asymptomatic mucosa carriage state to a major bacterial pathogen causing severe invasive infection. It is now a leading cause of neonatal meningitis and septicemia. Morphology of Streptococcus agalactiae. The cells of Streptococcus agalactiae are spherical or ovate gram-positive cocci of the size 0.6 to 1.2 in diameter. Cross walls during cell division are formed at right angles to the chain. Next, the organism is surrounded by a bacterial capsule composed of polysaccharide or exopolysaccharide. Next, the peptidoglycan present on the cell wall of Streptococcus agalactiae and the group B specific polysaccharide antigen is composed of rhamnose, N-acetylglucosamine, and galactose. The cell membrane had a lipid protein bilayer that helps in the transport of the different molecules.
In this topic, we will talk about the cultural testing conducted for the identification of the beta-hemolytic streptococci bacteria. The beta-hemolytic are divided into their designated Lancefield group, but for this discussion, we will focus on the important groups of beta-hemolytic streptococci, such as the group A and the group B. One of the bacteria that we will talk about is the group A Streptococcus beta-hemolytic bacterium, known as the Streptococcus pyogenes. To identify this group of species, Streptococcus pyogenes is isolated on blood agar plate. After 18 to 24 hours of incubation at 35 degrees Celsius to 37 degrees Celsius, growth is typically pinpoint for about 0.5 to 1 nanometer in diameter, and translucent, opaque, or clear in appearance, and white to gray in color. The colonies are surrounded by a wide zone of beta-hemolysis. Group A streptococcal colonies may appear either smooth and glossy or round and mucoid, which indicates the presence of M protein. For the cultural identification of the Streptococcus agalactiae, it is also isolated on the sheep blood agar. Colonies are medium in size, flat, entire margin, translucent or opaque, smooth, and non-pigmented. The medium is incubated for 18 to 24 hours at least 35 to 37 degrees Celsius. Most often, the colonies show narrow zone beta-hemolysis for about 95% to 99% of clinical isolates. However, non-hemolytic colonies also rarely may be observed.
For the next topic, we are going to identify the bacteria of beta-hemolytic streptococci and its biochemical reactions, which includes its basic characteristics, fermentation, and enzymatic reaction. To begin with, the first bacteria that we will identify is the Streptococcus pyogenes. Under its basic characteristic, it is negative in the following tests: the COMT test, the catalase test, the urease test, the VP test, and in 6.5% sodium chloride growth. On the other hand, it is positive in PYR test and is capsulated. Next will be the fermentation reaction of Streptococcus pyogenes. This includes the fructose, galactose, glucose, lactose, maltose, salicine, sucrose, and the trehalose, which are a positive fermentation for Streptococcus pyogenes. Moving on with this enzymatic reaction of Streptococcus pyogenes, it is positive in all of the following, such as alkaline phosphate, arginine dehydrolase, hyaluronidase, and neuraminidase. It is also variable in acetylene hydrolysis, which means it can be either positive or negative. For the next bacteria to identify, we will now focus on the Streptococcus agalactiae. For its basic characteristic, this bacteria is capsulated and is positive in oxidase test, VP test, and Camp reaction test. It is resistant under bacitracin resistance test, and as I mentioned earlier, it is a group B Landsfield group. Its pigment production is variable, which can be either positive or negative. For the fermentation reaction of the bacteria Streptococcus agalactiae, it is positive fermentation in the following, such as glucose, fructose, galactose, lactose, maltose, ribose, sucrose, starch, trehalose, salicine, glycerol, cellulose, and sorbitol. Lastly, we have the enzymatic reactions of Streptococcus agalactiae under acetone, alkaline phosphate, hyaluronidase, and neuraminidase, which have a positive reaction for the Streptococcus agalactiae.
So now, we will talk about the serologic test for beta-hemolytic Streptococcus bacteria. Serologic testing. It is an antibody test looks for the presence of antibodies, which are specific proteins made in response to infections. Antibodies are typically detected in the blood of people who are tested after infection. In this topic, we will talk about the serologic testing conducted for the identification of Streptococci beta-hemolytic bacteria. One bacteria we will talk about is the group A Streptococcus beta-hemolytic bacterium, also known as the Streptococcus pyogenes. Streptococcus pyogenes is an important global human pathogen that causes a wide variety of acute infections such as rheumatic fever. To identify these, we will use an anti-streptolysin O and anti-DNase B test, which is one of the most common of several antibodies that are produced by the body's immune system in response to an infections with group A Streptococcus. The anti-DNase test measures the amount of this antibody in the blood. It is typically done with the following anti-streptolysin O test. Another test to detect antibody to streptococcal antigen. The course of streptococcal infections, the extracellular products of the bacteria, acts as antigens to which the body responds by producing specific antibodies. Streptolysin O is one of those hemolysin produced by virtually all strains of Streptococcus pyogenes. The letter O indicates that this toxin is oxygen labile. This toxin has direct toxin effects on heart tissues. Next is the group B Streptococcus bacteria, also known as the Streptococcus agalactiae, is a type of bacterium that causes illness in newborn babies. In order to identify this bacterium, we will use the Welcogen strep B latex agglutination test. The Welcogen strep B latex agglutination test was evaluated as a method of detecting group B streptococcal antigen in urine, cerebrospinal fluid, and serum from neonates with early and late onset group B streptococcal disease. According to the ScienceDirect.com, urine is the best source of antigen, which was detected in 100% of 6 neonates with early onset group B streptococcal disease and 88% of 17 group B streptococcus infected neonates with urine available in the first 48 hours of illness. The Welcogen strep B latex test of the latex that compares favorably with a non-commercially available latex agglutination test.